So today, as Megan mentioned, we're going to talk about your Amplicon sequencing project and how to assess the data. And so I want to start by giving you an outline of what we're going to talk about today. So I'm going to start by giving you an overview of the Illumina Amplicon workflow. We're going to talk about important key concepts that we use for defining and describing sequencing data, especially in the Amplicon context. We're going to talk about the expected kit performance. I'm going to show you the options that we recommend for the analysis of your Amplicon project. And we're going to talk about different output metrics and the reports that are generated by the Illumina analysis platform and where to find them. So here's a summary of the Amplicon library preparation kits that we have available. We have several fixed panels with a defined content. They are all in the oncology field. So those include the TruSight Tumor 26, TruSight Myeloid, the TruSeq Amplicon Cancer Panel, and TruSight Tumor 15. We also have a custom solution through the TruSeq Custom Amplicon Kit. We abbreviate this kit with TSCA, so you might hear me mentioning it with this abbreviation throughout the talk. And we have the 1.5 version and then our newer low input version. This is a kit where you use our free online software design studio to enter your desired target regions to make up your own Amplicon kits. I also have a link here to the 16S metagenomics webinar. So 16S is also considered an Amplicon, but it's a little bit different compared to our other products. It has its own specialized analysis, and we have actually a recorded webinar entirely dedicated to 16S. So just follow the link here on the slide if you would like more information about that. So this slide is a summary overview over our fixed panels for oncology. This is taken from our Library Prep and Array Kit Selector tool. There's a link here at the bottom of the slide. And I just wanted to give you an overview over the key differences between these four kits. The kit selector in general is a great tool for you to help you decide which panel might be the best for you. So the panels vary in terms of key use, so the targeted regions, the amplicons that are contained in each of the four kits. The table also lists the time that the library prep takes, the genomic content size, and things like input amount and whether or not it is FFPE compatible. You can also, from the kit selector, directly go to the product page and the support page of each of these panels to get even more information. So let's take a step back and talk about when an Amplicon workflow might be right for you. So anytime you're not interested in the whole genome, but only want to focus on specific regions, and we call those our targets. So here in this cartoon, you can either take your whole DNA that you put into your library prep, make all of this into a sequencing library, or you can decide to zoom in on specific regions, such as shown here in red. That will be the case for an Amplicon workflow where we are not wanting to look at everything, just specific regions, and we need to find a way to only highlight those in our library prep. So the key is that our target regions are precisely defined, so we need to know what we're looking for. And for the Amplicon approach, we're using probes that synthesize the target regions and thereby convert them into a sequencing library. And that is the key difference to an enrichment type analysis. For enrichment, this is also something where we're looking at specific targets. However, we are using capture probes to pull down the target regions. And that requires also different considerations when it comes to the analysis. So again, we have a recorded webinar entirely dedicated to this, and I'm providing a link for you here on the slide. So let's go over a couple more details of the Illumina Amplicon library preparation. So we're starting with probes that are called the upstream locus-specific oligo, or ULSO, and downstream locus-specific oligo, or DLSO. So those probes hybridize to flanking regions of our genomic DNA, and then an extension and ligation reaction occurs between the probes on the same strand. So they hybridize on the same strand, and then through the extension ligation, they are joined together to form the amplicon. The next step is then a PCR to add indexes and the remainder of the adapter region. And that gives us a uniquely tagged amplicon library that is now ready, has all of the elements that we need for cluster generation and sequencing. So sometimes your target region might be larger than what can be spanned with one amplicon. So what happens then is that neighboring amplicons are going to be designed on opposing strands, such as shown in this cartoon here. So that the probes do not interfere with each other, you'll have the first amplicon on the plus strand, the second on the minus, the third on the plus. So they alternate in order to span your entire target region. So some of them will sequence the plus, some of them will sequence the minus strand. There might be situations where you are interested in sequencing both the plus and the minus strand. And this can be really important if you have FFPE samples. The FFPE process itself can yield artifacts through deamination or other events that can cause single nucleotides to change. And so we can distinguish these from true variants by doing a dual pool design, which some of our kits offer. So what you're going to get is a mirror image of your original oligo pool that's going to have the same amplicons but on the opposite strand. So together with pool A and pool B, you're going to sequence your entire target region, both strands, and you can confidently distinguish these variants from any artifacts of the FFPE process. I want to highlight the TrueCyTumor 15 workflow because it is a little bit different compared to all the other amplicon workflows that we have. So here, in our first step, we have primers that actually anneal in a traditional PCR. So we have a forward and a reverse primer. Those are generating our amplicon. This again has to follow a tiling method that uses 2 oligo pools for this giant multiplex PCR. So again, we have a pool A and a pool B. That's a bit different because the 2 pools now actually are going to not be mirror images, but they each will have their own target and collectively will span your entire target region for the TrueCyTumor 15 panel. The next step is a second PCR that now adds the indexes and the adapter regions. That's very similar to the classic workflow for Amplicon. And again, this gives us our uniquely tagged Amplicon library that's ready for sequencing. Okay, let's switch gears a little bit and talk about important concepts that we need to know to understand sequencing metrics. So the first thing that I want to talk about is the term of mean coverage and mean read depth. I'm sure you've heard this before, a very common term in sequencing in general. When we talk about amplicons, the mean read depth is calculated across all of our sequenced on-target amplicons. So I have a number example here. So we have 4 targets in this example. We have different numbers of reads for each. Target 1 is 500, target 2 has 1,000, target 3 has 50, and target 4 didn't get any reads aligned to it. So in this case, our mean coverage would be 508x derived from these 4 targets. Another term that comes up very frequently is the uniformity of coverage. This is another concept that we use to evaluate our data, and that is the percentage of amplicons that have a coverage of greater than 0.2 times or 20% of the mean coverage. So in this example, with our mean coverage being 508x, if we do the math, 0.2 times the mean coverage is 101.6, and then we have two targets that are above this value. So that would give us a uniformity value of 50%. And then we'll often talk about the specificity. Now that is the percent of passing filter reads aligning to the defined target. So if you have any reads aligning somewhere else in the genome, such as here in the region shown in blue, that would decrease our specificity percentage. So how are these off-target amplicons generated? Let's talk a little bit about that. So here's an example of two on-target amplicons. Those are exactly what we are designing, what we want, what we expect. So you have amplicon number 1 that is formed of the ULSO-DLSO pair 1, and amplicon 2 formed by ULSO-DLSO. Now what can happen, there's multiple examples for off-target amplicons. It could be that that same pair for amplicon 1, so ULSO-DLSO pair 1, hybridizes somewhere else in the genome, and that is usually going to be a region very similar in sequence to the one that we targeted. So that is one example for an off-target amplicon. Another example is that, for instance, ULSO2 unspecifically binds somewhere else in the genome. By itself, it wouldn't be able to form an amplicon, but if by coincidence a different DLSO, so here DLSO1, hybridizes in the vicinity of ULSO2, they can together form an off-target amplicon. Now we have an unexpected probe pair that can form one of these off-target amplicons. So this can happen. Typically, those amplicons are not going to be very detrimental to your analysis. And then one more term I wanted to bring up here is the dropout. So dropouts are target regions that have zero coverage, so no reads at all. For instance, in this example, that would be our target 4. Okay, so now what are our specifications? That is an important concept when you are starting to analyze your data. If your data looked good, to come back to these specifications. Here for our 4 fixed panels, you see that the specifications are different for each panel, so they vary by panel, and you'll see words that we just discussed. So they will contain, for instance, a certain mean coverage value, a minimum coverage, specificity, or coverage uniformity. These are all terms that we use to assess our performance. And something else that I have listed here in this table is the number of samples per run that are recommended, because this, of course, directly ties in with the coverage to be expected for each kit. You can find more information also about these specifications in the respective kit data sheet. Now, how about the TrueSeq Custom Amplicon? Well, it is a custom solution, something completely new that you're designing. So we don't have any specifications per se, but we have performance guidelines. Since, of course, you're using the Design Studio software to create that design, it's an algorithm that takes a lot of considerations into account to give you the best possible design that is going to be successful. And so we are expecting a specificity of higher than 70% and a uniformity of coverage of higher than 80% for these panels. All right, so now let's talk a little bit about the manifest. That is something that's used in the analysis for Amplicon workflows. It is a text file and it contains target information. It's also used for the alignment. We often get the question, where do you find your manifest? Well, for the commercially available panels, it is available on our support page under the download or product files section. And for the custom panel that you create in Design Studio, it will be available directly in the Design Studio software under the download files section. But you have to keep in mind that the manifest will actually only be generated after the probes have been synthesized and are delivered to you. So all this information that we're going to go over in the next couple of slides, the detailed information about the probes, is actually only going to be available really after you have finalized your project and ordered it. So keep that in mind when you're looking for your manifest. The panels require either one manifest, so that's the case for the single strand TrueSeq Custom Amplicon and TrueSight Myeloid, or two manifests. That's the case for TrueSight Tumor 26, TrueSight Tumor 15, and the TSCA dual strand design. And the manifests are divided into two sections. We have a probe section that talks about the ULSO and DLSO sequences in detail. And then we have a target section that talks about the position and the sequences of the target region. And to keep in mind that the manifests are similar for the different kits, but there are some sections that are not exactly identical. So when you're looking at a manifest, just keep that in mind. So you might see something like a header section in one or the other manifest, additional target IDs or things like that that are a little bit different. But I'm going to talk to you right now about sections that are going to be present in every manifest. So here's our probes section. Here I'm highlighting the target region coordinates. So that's the start and the end position of your target. Then here we have the ULSO and DLSO sequences. So the detailed sequences of those probes that we talked about, they are here shown without adapters. So these probes are going to have an overhang. There's one section that hybridizes with the genomic DNA, and then there's a little overhang stretch, which is a part of our adapter for sequencing, and that part is also used in the subsequent PCR reaction to prime off. But what we need here, what's important, is the part that binds to your target, and that's what's shown here. And then in this table here where it says strand, that is the strand on which the ULSO, DLSO is designed. So now let's move on to the target section. That is a little bit lower on your manifest, so you want to scroll down a little bit. You'll see here in this example, it starts at position 440, so a little bit further down. That section has your Amplicon coordinates, so that includes the probes. That is where your individual Amplicon starts and ends. And then we have the sequence. This is also a very important section here. This is our targeted sequence without our probes. So this is essentially the part that is added by extension ligation between our oligos. And this is also the part that is used for variant calling. You have to keep in mind that our probes are hybridizing. They are not created by synthesis. So that part cannot be used for variant calling. So it's only the portion in between that is key to finding your sequence variant. Here we have our ULSO, DLSO ID combination. So those are like unique names that are given to the probes that we can use later for the analysis. And then here we have the strand to which read1 maps. One more thing in the target section I want to draw your attention to, this column that says target number. Now in the beginning of the target section, that number is always going to say 1. But as you scroll down a little bit lower, again towards the bottom of the manifest, you'll see some higher numbers pop up, such as 2s or 3s. Now all of the 1s are our on-target amplicons that we expect that are generated by ULSO, DLSO, by an expected pair. But then we also have some off-target amplicons, like we discussed before. So some of them will be allowed in your analysis. They are present, they are not detrimental. If we didn't allow any off-targets, it would be very difficult to design any probes for these kinds of scale projects. So we are taking them into account in our manifest, and they are shown here with the ulysol-deulysol pair and with these numbers that are higher than a 1. Okay, so now let's talk about your actual sequence data and how to assess it. So the first question you're going to ask is, how should I go about analyzing my data? So the Amplicon workflow is supported on the MiniSeq, the MySeq, and the NextSeq. And you can perform the analysis either locally on your MiniSeq in LRM or Local Run Manager. You can also do it locally on your MySeq in MySeq Reporter or MSR. Or you can do it on the cloud on our BaseSpace Sequence Hub. This table summarizes the recommended analysis options for each of the kits, for each of the Amplicon kits. You'll see I'm listing it out here for LRM, for MSR, and for BaseSpace. These are all very similar apps, depending on where you are. If you're on the MySeq or the MiniSeq, they all have similar names. But keep in mind that these are separate analysis solutions. So they might not be completely identical, and each of them also has its own workflow and its own reference guide. So for TrueSight Tumor 26, we're going to want to use the Amplicon DS module on LRM, workflow on MSR or app in BaseSpace. For TrueSight Myeloid and the TrueSeq Amplicon Cancer panel, it's going to be the Amplicon or TrueSeq Amplicon solution. TrueSight Tumor 15 has its own analysis solution with its own name, TrueSight Tumor 15. And then if you are using the TrueSeq Custom Amplicon solution, you're either going to use the Amplicon or the Amplicon DS solution, and that depends on whether or not you've chosen a dual pool design. This is one of the first questions that the Design Studio software is going to have you choose when you're doing your design. So based on that, you're later going to select the appropriate analysis. And I wanted to mention also that all sequencers have the option to just generate path cues on the instrument and then do your analysis in BaseSpace. So what are the important metrics that we are now going to take a closer look at? So we've defined them earlier in the talk. So we want to look at coverage uniformity, and specificity. But then of course there's other key considerations such as individual Amplicon performance. You want to see where you have potential dropouts, how are your Amplicons doing. And of course there are our variants. That's probably why you're doing this analysis in the first place. So in the next section of the talk, we're going to look at where you can find all of these metrics. So let's start with BaseSpace, specifically the TrueSeq Amplicon and Amplicon DS modules. They are very similar, so I'm showing them here together. And the first thing I want to draw your attention to is the coverage summary. So you'll see on the left-hand side here that where we are in BaseSpace, we are under the analysis reports in the summary section. And here you can see the coverage summary, so the Amplicon mean coverage depth for each of the samples in this project. For the coverage uniformity, you want to go to your individual samples. So this is what it looks like in BaseSpace here. And here you can see the uniformity of coverage value. It also gives you this nice graph where it shows the Amplicon mean coverage and the uniformity of coverage thresholds. So there's this red line shown, that is the low coverage threshold. So that is at the 0.2 times Amplicon mean coverage value. And all of the blue points are going to be above the threshold, and all of the red points are going to be below. And this is pretty interactive. You can actually hover over individual points and it shows you exactly which region they correspond to and what the coverage is for that data point. And finally, this data can nicely be exported into Excel if you want to save it somewhere else on your computer. Now for the specificity value, we want to go back to the summary section, and that is shown here, the percent of aligned reads. You'll see typically for our Amplicon values, that's going to be very high, higher than for instance, for a typical enrichment analysis quite often. So this is where that value is again summarized for each of the samples in your project. Now for your variant data, again in the summary section you find just a summary layout for your small variants, also insertions and deletions, just as an overview of what kind of variants were identified in each of the samples and how many. And then if you'd like more details, you can go to the individual samples to dig a little bit deeper in your analysis. Now let's look at TrueSight Tumor 15, the coverage and specificity, also available under the analysis summary section, under the analysis reports. And you'll see here this is shown for Pool A and Pool B separately, and it's actually shown as a percentage value of everything above 500x coverage. And then on the right-hand side, you'll see the percent on target, again for Pool A and for Pool B. TrueSight Tumor 15 also gives you an analysis report that contains the variants and the no-calls for a specific subset of variants that are contained in the report definitions file. And I'm providing a link here to this file. So this is what that report looks like. It's in PDF form, and it is essentially a summary that goes over specific variants, what they are, what the frequency is, something that you can download, use, very nicely summarized. And this is what the report definitions file looks like. So that's where you can go if you want to get even more detail of where the position is of these variants and what the reference is, what the alternate is, to get a better idea. But of course, we have variants in everything that's contained in your target. So for reporting on any other regions that are in the manifest, but perhaps not in the report definitions file, you want to refer to the output files that are generated by the app for further analysis. So what are those output files? Let's talk about them. They can be very important. So we can use those to do our own calculations for performance metrics. They are generated by all our analysis solutions, so LRM, MSR, and base. And some of the important files that I want to talk about today are the coverage.csv file, the .bam or bam.bai file, and the VCF file. If you've ever worked with us here at Tech Support, you may have been asked to provide us with these files. We use them often for our analysis and to help you understand your results better. So where do you find those? I wanted to give you an overview here on this slide. So in Local Run Manager, of course, you're also getting the PDF summary report for each sample, very similar to what we just looked at in base, but it also generates these important output files, and they can be found in your alignment folder in the Run folder. For MSR, MySQL Reporter, the output files are going to be found in your analysis folder, and here I'm providing you with the file paths to essentially that lead you to those files. And then for TrueSight Tumor 15 in MSR, there are 2 additional files that are generated. So you're also getting that PDF summary report that we just looked at from the base app. So MySQL Reporter also generates this file. And it generates a sample metrics report.txt file that contains the coverage and specificity metrics, just like the base app. And then in BaseSpace, those files can be seen here under the Output Files section. So if you remember earlier, we were under the Analysis Report section. Now we're under the Output Files. And that's where you can see listed for each sample the BAM files, coverage.csv, VCF, and other files that can be downloaded and used for further analysis. So let's take a closer look, starting with the coverage.csv file. So this is a great file for analysis. It essentially just contains your read count per sample, per amplicon, and the name of the amplicon in the first column. So this can be used to calculate your specs. So you can calculate the mean coverage, the percent amplicons below a certain coverage, for instance, and the coverage uniformity and others. So in addition to what the app calculates for you, you can also do your own calculations with this, especially if you have, let's say, a certain coverage that's important to you or your lab. You can calculate what percent of amplicons are below or above that. So I want to walk you through this calculation. So to get the mean sequencing depth, you just add up the number of reads that you have and then divide them by the number of amplicons. And you can either do this sample by sample, or you can just collectively do it for your whole run, for all of your samples. The next thing that you want is that uniformity cutoff. So that's 0.2 times the mean that we calculated under 0.1. And then to get the uniformity of coverage, you want to take the percentage of amplicons that have a coverage greater than that cutoff calculated under 2. And then the coverage.csv file also has our off-target amplicons. So very similarly to the manifest, all the way at the bottom, you'll find some amplicons that are ending in a number higher than 1. So you'll see in the beginning, although we have different names, everything ends in a .1. That's all on target. A little bit further down, you'll start seeing things popping up, perhaps, that are ending in a .2 or .3 or higher. These are off-target amplicons, and you'll even see the number of reads that are associated with those. So those take away a little bit from our specificity, but you'll see we can clearly separate them out often, and it is something that is taken into account in our analysis. So let's talk about BAM files. The BAM file contains the sequence alignment data. This is what is used as input for variant calling. And it can be useful for seeing more details on how the alignment proceeded. And also something that we might want to look at when we're assessing the variant calling. So for these files, we need a tool to visualize them. You can use tools like IGV or JBrowse or UCSC Genome Browser. And I'm showing a screenshot here for how a BAM file looks like when it is shown in IGV. You see your individual reads, how they are aligned to the genome, that's shown at the bottom. You can even see the direction of the read. You'll see different variants shown here. So just to get a visual of how the alignment proceeded. If you are looking at these files in a tool such as IGV, you'll also need the bam.bai file that I mentioned earlier. That's just an index file. It's typically a lot smaller than the BAM file. And wanted to provide one more reference here for more details on this file format. Now talking about the VCF or the GVCF file. So VCF stands for Variant Call Format. This file is created by our variant caller. It contains meta information lines, also a header line, and then it'll have the data lines that each contain the information about a position in the genome. And so now the G in GVCF essentially just stands for genome. So those are very similar to the VCF file, but just contain additional information. So in addition to our variant calls, you also have all the calls that match the reference. Sometimes when I go to the GBCF file, it's because perhaps I was expecting a certain variant, I don't find it in my VCF file, then I go to the GBCF just to see what was going on at that position. And these files can be opened either with, for instance, with Illumina Variant Studio or with Space-Based Variant Interpreter. And I just want to give you an example here what a file like that looks like shown in Variant Studio. So these are essentially visualization and filtering tools. So you'll see Variant Studio here shows you all the information about the variants from the VCF files. So the gene, the actual variant, what it is, the chromosome, coordinates, classification, and so on. So a great way to filter, and we have an entire webinar dedicated to Variant Studio, and I'm providing a link here at the bottom of the slide. Space-Based Variant Interpreter, essentially the same idea, also for report generation, filtering, and visualization of our variants from the VCF file. This is just a screenshot from the Variant Interpreter. And again, if you're interested, we have a webinar that's recorded, available for you to review at any time, and I'm providing a link again here at the bottom of the slide. Now talking a little bit about UCSC Genome Browser. This is a tool that can be used to assess Amplicon performance, even predicted Amplicon performance, so even before you've actually done your experiment. Because you can look at GC content, any underlying SNPs for where your Amplicon is, any repeat regions. And in fact, we are providing links directly to UCSC Genome Browser from our Design Studio software. So you can go under Targets in Design Studio. Here is a little link that takes you to UCSC Genome Browser to that particular target region. And this is what it looks like. So you'll see in orange or yellow, that will be your target region as you have defined it in the TrueSeq Custom Amplicon workflow. And then here in green, you'll see the Amplicons that the software has designed. What I think is really neat is you can see there's a thick part of the Amplicon and then a thin part. The thick part corresponds to the probes, and then the thin part is that region that is added through extension ligation between the probes. What you'll also see here in this particular example, the orange region is larger than what can be spanned by one Amplicon. So you'll see there is some tiling going on, so you have two neighboring Amplicons. And as I mentioned before, the region between the probes is what's used for variant calling. So it is actually designed in a way that you have a continuous region spanning your Amplicon between two probes, so between two Amplicons. So the Amplicons are kind of tiled a little bit in that sense. So here you can filter for tracks such as GC%, the repeat master track that looks at repeat regions, common SNPs, and you can just see how difficult might it be for an Amplicon to be synthesized from that region. We know that things like an abnormal GC content can make Amplicon performance more difficult. Of course, these are all things that the Design Studio software takes into account, but it gives you the option to visually inspect for yourself to see how the Amplicon design went. Okay, that brings us to the summary for everything that we talked about today. So we talked about key concepts, definitions, terms, what they mean, how we use them. We also talked about choosing the appropriate analysis workflow, the recommendations that we have at Illumina for each of the kits that we provide. Then I've shown you where you can find and how you can evaluate the performance metrics for each kit. And then also how to use the output files to further analyze your data if desired. It's always important to know what are the expectations and why do we have them. This is something that we always want to think of when we are trying to decide whether our project went well and what are the metrics that we want to really focus on. And then also I encourage you to compare your data to the expected values that are provided either in the slide deck or also in the respective kit data sheet. Another great resource I want to leave you with here is the public data sets on BaseSpace, because in addition to any fixed specifications, sometimes you just kind of want to see how does your data compare. And that's what we have public data sets for on BaseSpace. We usually have some information about the type of sample that was used for the run for the project. So at any time you can go to that public data part, browse through the project, look at different metrics, see how that sample type compares to your sample, and then draw conclusions based on that. And then on my last slide, I just wanted to give you a couple of resources that you might find useful. Of course, tech support also should be your resource anytime. Give us a call, any questions that you have about your analysis, about your experimental design, about Design Studio, anything, we're always there to help. And with that, I'm happy to take any questions.
All right, thank you, Judy, for that very informative webinar. So we will enter the Q&A portion, and just a quick reminder to please type your questions into the chat window. Okay, so first question, is there a minimum read length for my Amplicon?
So there is, when it comes to the analysis, some of the workflows do have technical limitations. So for instance, the TrueSeq Amplicon and the Amplicon DS workflow have a minimum of a 50 base pair read length. But besides that, of course, we just want to make sure that we choose a read length that's appropriate for your project.
Okay, what's the percentage of spike-in that I should choose for Phi-X?
That's a great question. So with all of the panels that we really talked about today, these are all, even though they're Amplicon, we have a large number of different Amplicons. So these would all be considered high diversity. So none of those would be a single Amplicon. So for that, we just would go with a 1% Phi-X spike-in that's recommended for instrument QC, but not required. So the short answer is you don't need any. If you have other Amplicon projects, such as you have designed one Amplicon that's going to be low diversity, then we want to spike in some Phi-X, and that depends on the sequencer. So if you have a specific sequencer, feel free to type that in the chat or call us at tech support. We can help you with that.
Okay, great. When should I choose Amplicon over enrichment?
Great question. So the Amplicon workflow is usually something that is used for smaller projects. For instance, for TrueSeq Custom Amplicon, we have up to 1,536 targets, or 650 KB of total target size. Enrichment is often a great solution for larger projects. So if you are above 500 KB all the way up to 15 MB megabases, that's when we want to do typically an enrichment solution.
Okay, another question, is there a suggested tutorial to learn more about BAM files?
Yeah, so ITV is a great solution to kind of go and play a little bit with the BAM file and look at different metrics. It is a third party, so we want to look at the Broad website to find potential tutorials, but that's where I would start.
Okay, great, thank you. Another question, can I directly compare data analyzed on the MySeq with data streamed to BaseSpace?
So the apps on BaseSpace are similar to the workflows in MSR on the MySeq, but they're not identical. So these are separate analysis options. They each have their own settings and own workflow guides. So they are similar, but you want to refer to making sure that you chose the right settings and keeping in mind that these are separate workflows.
Okay. And what algorithm do the workflows use for alignment?
So for the alignment, we use the Bennett-Smith-Waterman algorithm in the targeted region.
Okay, and which variant collar is used?
So for the variant collar, we use the Illumina developed somatic variant collar.
Okay. What controls should I consider when running any of the Amplicon panels?
So when it comes to the controls, the important question is, what type of control, right? So for the TrueSeq custom Amplicon, for instance, there is a control DNA and a control oligo pool provided in the kit. So this is something that you can use to just test your library prep itself, right? That's one type of control. Other types of controls could be DNA that has a very specific defined variant. So for instance, Horizon might be selling controls like that have variants that are defined that you could run through the library prep and then the analysis. So these are different options, different types of controls, depending on what you're looking for.
Once a custom TSCA library has been sequenced, what is the typical percentage for undetermined reads?
So that's a good question. The undetermined reads, they usually will be things like your PHYX that goes into the undetermined FASTQ file. So that will directly depend on how much PHYX you spike in, right? If for a typical run where there's nothing wrong, everything went well, it should correspond somewhat to the percentage of PHYX that was spiked in. But then, sometimes if something went wrong, if for any reason reads cannot be identified and not demultiplexed with the index read, they would go into the undetermined. So if you feel like you have an unusually high value, something that maybe is a lot higher than what the PHYX that you added, then maybe you want to give us a call. We can take a look to see what happened. Maybe the reads were just thrown in there because for some reason they couldn't be associated with the index that we entered, and that can have a variety of different reasons. Sometimes we can fix that. So give us a call.
All right. We have time, I think, for one last question. What's the advantage of the ligation extension approach?
That's a great question. I would say, both workflows are something that we have established. There wouldn't essentially be a clear advantage of one over the other. It's just two different ways to do this library prep. So we have the extension ligation approach on one end, where we can put all of the oligos really in one pool, and then we have the TrueSet Tumor 15 workflow, which is just a different technique. Both of those are great techniques. Just that, in biology, often there are just different techniques that we have to come to the same goal. So I would say both of them are great options.
Okay, great. Well, thank you very much, Judy, and thank you again to all of our attendees.